Okay, so what I'm going to do today is go through the homeostasis review sheet that we did, or that you're supposed to have done for today. I'm just going to go through the answers, kind of type them in so you guys can see what they are. And so I'll go through kind of step by step. There may be some pauses as I'm going, or I'm pausing the video to go look at something else quick or bring a picture up. But I'll try and keep that to a minimum. Alright, so when we're looking at the homeostasis review, or looking at for the test tomorrow, so one of the first questions here is looking at the types of passive transport. So there are three types of passive transport that we discussed or talked about, or we didn't discuss, but that were in the videos as well as the lessons. So the first type, and actually let me change the size of this to make it easier for you to see. And we'll go, there we go. And so the first type is diffusion. And so remember, diffusion is just simply the movement of particles from an area of high concentration to an area of low concentration. The next one was what we call osmosis. Osmosis is kind of the same thing as diffusion, but remember it's basically the movement of water molecules through a membrane, right, from high to low concentration. So it's going from where there's a lot to where there's less. So diffusion, an easy example, is like putting a drop of water in a glass and watching it basically spread out. It's going to diffuse. Osmosis is the same thing, but it's going until it's the water going through a membrane. The last type is what we call facilitated diffusion. Okay, so again, this is the same thing as diffusion. It's always moving from high to low concentration. And then facilitated diffusion means it's going through a protein. So when we're looking at kind of the cell membrane, we can see those proteins that they travel through, and we'll bring that up later when we talk about the structure, composition, and function of the cell protein. Okay, so the next one says just explain what a concentration gradient is. So when we're looking at a concentration gradient, so this is basically the it's the arrangement of particles in a solution. So what we're talking about, is basically again the concentration. So we're looking at an example. So the concentration gradient, in this case, what we have is we have a very low concentration over here on the left, very high concentration on the right. Whereas you go from the kind of right to left, the concentration decreases. So concentration gradient is just kind of that arrangement. So high over here, low over here. Eventually everything wants to get to what we call equilibrium, where everything is kind of equally spread out. So if we're talking the containers, it goes from right here to here, right? This is what we call equilibrium, where everything's kind of equally spread out. Here we have a high concentration and a low concentration. So you can kind of see those changes. But the concentration gradient is just simply how those particles are arranged in a solution. Yeah. So the next one is looking at how osmosis is related to diffusion. Yeah, so what we're looking at here is basically osmosis. So osmosis is diffusion of water through a membrane. Okay, so again, looking at this, and this kind of is an image. Yeah, so you can see here with diffusion, it's simply the particles are simply spreading out until everything is equally spread out, or we get to equilibrium. With osmosis, what we're looking at is we're looking at the movement of water. So like the solutes are simply too big. So you can see in this picture here, the particles on the right are very tightly packed together, where everything over here is we have that kind of low concentration, they're kind of spread apart. So what the water will do is the water will move. Remember, this is a membrane here, so water doesn't just freely move. Right? It's not kind of even. Right? So what we're looking at here is the concentration will eventually get to where they're equal to each other. So almost the red dots here are all equally spread out now, right? But the water moved from the left side to the right side to allow that to happen. So again, diffusion is simply the spreading from a high concentration to low concentration. Osmosis is doing that in order to get everything equal, right, through a barrier. Okay, so here. Okay, so looking at the definition for hypotonic, hypertonic, and isotonic solution. Yeah, so what we can see here is hypertonic is a solution where there's a higher concentration of solutes outside the cell, so water wants to move out. So remember, we want things to be equal, so you can see right here in the cell, they have the 4 black dots here representing sodium, Where on the outside we have 1, 2, 3, 4, 5, 6, 7, 8. So what water wants to do is water wants to move outside of the cell because it wants to get the kind of everything equal to each other in percentages. So you can say here we have, let's say it'd be 50% water, percent solutes. Right, outside, since it's only 30% water, water wants to move outside. Okay. So then we have a hypotonic solution, is basically the concentration is higher inside the cell. So if we're looking over on the right, we still have the 4 on the inside, but only 2 outside. So because that water wants to move into the cell to try and spread these out a little bit further to be equal to the outside. And so kind of one thing I always remember is hypertonic is things want to get, the water wants to get out because it's hyper. It has a lot of energy, so it wants to move out. Hypotonic is kind of low, so it wants to go and kind of move into the cell. And whereas isotonic then is there's an equal concentration of solids both inside and outside. So you can see there's 4 outside, 4 inside. So water is still going to move, but it's going to move equally into and out of the cell. And so that's going to look at the definitions for hypertonic, and isotonic solution. Okay, so when the ink is dropped into the beaker of water, what's going to happen is the ink will spread out until it's evenly dispersed. Alright, so in that process is called diffusion. So what I have here is I put a video in there so you can kind of see it happening, kind of showing what it is. But the particles will simply spread out. Okay, so looking at the differences between passive transport and active transport. And so there's kind of one key or two key differences. Alright, the first is that active transport requires energy. And so active transport requires energy to happen, so in order to kind of work, it needs something to power it, right? Whereas passive transport just happens, there's no energy required at all. The other kind of key difference is the movement. And I'll pause to type this in quick to save some time. Okay, so looking at the differences between active and passive transport. So key things are active requires energy, where passive there's no energy required. So passive is just kind of free movement, where active energy or active transport requires energy. Active also goes from a low to high concentration, where passive goes high to low. So you can see with passive transport, we have our high concentrations on the left, low on the right. So they move naturally from that high concentration to that low concentration. So whereas active transport is using energy to go in the opposite direction. So we're going from a low concentration being pushed into a high concentration. So those are kind of those two key differences between active and passive transport. Okay, so the next question is, describe the structure, composition, and function of the cell membrane. Sorry, I almost missed a question. So first one going back is number 7. What does it mean for a solution to be in equilibrium? So equilibrium is just when solutes are equally spread out. So when we're looking at kind of an example, you can say an isotonic solution is in equilibrium because they're equally spread out. We have 4 outside, 4 inside. Whereas a hypertonic and hypotonic solution, they're not equally spread out. So equilibrium is when basically everything is equal. So when both sides are equally to each other, it is in equilibrium. Okay. So now I'll set the answer for number 8. Okay, so when we're looking at question number 8, so the structure, one of the structures we call a phospholipid bilayer. So when we're looking here, we have the layer, we have one layer on top, and then on the reverse of that, we have kind of that bottom layer. So there are two different sides, kind of form almost like a sandwich, where you have the tails on the inside, and then the heads on the outside that are hydrophilic heads, which are light water, and hydrophobic tails. So that was back from the Bioman game that we went through. Remember the composition then is kind of what it's made of. So it's primarily made of those phospholipids, which are those kind of individual pieces. There's a big variety of proteins. So we have the glycoproteins help the structure. Again, proteins help hold together. We have like our protein channels to let things in and out. Now we have the what's the other one at? So individual proteins again here that help hold things together. Remember we also have like the cholesterol. That's on the inside of the cell membrane that again helps stabilize the structure and help hold everything together. Right? Okay, so then the function, or the whole kind of main thing is it protects the cell. Right, so we also have that selective permeability where only certain things can get in and out. So it allows things that our cell needs to get into it while blocking others. Remember, like water can go through these protein channels, or like the aquaporins, whereas other chemicals can't. Right, and I think really small things like carbon dioxide and oxygen can just go through the membrane. All right, but again, that select permeability allows the cell to take in what it needs, right, and prevent other things that are the bad things from getting into it. Okay. So again, with what does it mean for a cell membrane to be semi-permeable? Let's do this one real quick here. So remember, if it's semi-permeable, that means it only lets certain things into the cell. So we're looking at here, the cell membrane here, it's going to block a lot of things that the cell doesn't need, right, or would be harmful to it. So again, we have those pores that allow certain things into, right, the cell. And it also allows certain things out. Okay, so again, looking at the questions, 10, 11, and 12. So the purpose of proteins that are found in the cell membrane, such as aquaporins, or you can kind of see here the protein channel. What they do is they simply let in particles in the cell that are larger. So water can't move through the cell membrane because it's too large. And remember, we also have the tails on the inside that we call hydrophobic that don't like being around water. So if water were to get in here, it'd be a really bad thing. So they need to have a way to pass through the cell membrane without coming into contact. So they can travel through this channel here that allows them to move through. So it also lets through larger particles. A lot of times we're looking at, say, sugar, for example, is a large particle. There will be proteins that allow that into the cell. So then we have the gated channels, and gated channels normally would be just like this, but there's some sort of signal to allow them to open. Otherwise, they're closed. So the signal could be energy, or it could just be simply some sort of particle attaching to it. So like sodium ions, for example, can only get through the cell membrane through a gated channel. So it's called a sodium-potassium pump. But gated channels is basically, they open and close in response to certain signals. Okay. So then looking at question number 12 again here, so it says 3 red blood cells are placed in hypertonic, hypotonic, and isotonic solutions. So looking back at question 4, we want to look and see what happens. So we can come back here. So that's why I included this picture here. So if you take a blood cell and put in a hypertonic solution, right, water is going to leave the cell, so it's going to shrink because the concentration of salt is higher outside. So water wants to move out to get that equal. All right, hypotonic, water is going to move in to get that concentration equal, so the cells are going to swell up and get larger. Now, whereas in isotonic, it's going to be equal movement into and out of the cell.